Good afternoon, students. Welcome to the world of biology. Today we will start the part number 4 of chapter number 5, and today's topic is linkage. First of all, what do you mean by the linkage? Basically, it is a close association of genes on the chromosome. When suppose this is the chromosome and these are the two genes here. This is gene number 1, this is gene number 2. These two genes are known as linked genes, and its inheritance will show the linkage. First of all, linkage word was proposed by Morgan, and its detailed study was carried out by Batson and Punnett. In the fruit fly, these scientists used the fruit fly for the experiment, and the name of fruit fly is Drosophila melanogaster. So, what is the linkage? What are the important facts about the linkages? What was the experiment on Drosophila? Everything will be discussed here by using the PPTs. So, if we discuss about the linkage, as I have already told you, linkage is the close association of genes or other DNA sequences on the chromosome. As far as the closer two genes are to each other on the chromosome, the greater the possibility that they will be inherited together. So what will you see when two genes are closer to each other on the same chromosome? Then there is the more probability of the inheritance together in the next generation. The independent assortment is absent where linkage is present. This is the important fact. Where the linkage is present, there will be the absence of independent assortment. The distance between genes is inversely proportional to the linkage. You can see the relationship between linkage and the distance of the genes. Linkage is inversely proportional to distance of the genes. You can see the fact what is the gene linkage. You can see this is the gene 1, that is on the top of the chromosome number 1, and this is the chromosome number 2, where these blue, light green and this highlighter color can be seen, where 2 and 3 and 4 these genes are present closer to each other, and this is known as linkage group. So gene 1 is unlinked gene. Unlinked genes are located on the different chromosome, and in the second part you can see these are the three genes which are closer to each other, and this group of genes is known as linkage group. A set of genes at different loci on the same gene except for crossing over tend to be inherited together. The next is Morgan's experiment on Drosophila. Basically, Morgan performed a dihybrid cross by using the Drosophila, and he selected two traits of Drosophila. First of all, we will have to discuss about the parents. The first parent male, he was taken as a brown bodied and red eyed. The body color of the male insect was brown and the color of the eyes were red. What about the female? The body color of female was yellow and the color of eyes were white. It is shown by small y, capital W, capital W. On the other hand, the dominant character, that is YYWW. He expected Mendelian result in the cross. He was expecting the Mendelian results, but results were unfortunately different because the Drosophila show a linkage for this character. He was expecting the Mendelian results, but the results was very different from this. This was because of the presence of linkage, because these two genes are linked genes and present closer to each other. Morgan hybridized yellow-bodied, white-eyed females to brown-bodied, red-eyed male. It is considered as a wild strain, and intercrossed their F1 progeny. He observed that the two genes did not segregate independently of each other, and the F2 ratio deviated very significantly from 9:3:3:1. What do you mean by this? Basically, if that was the Mendelian cross, then the result were 9:3:3:1. but results were different from this ratio. You can see the figure here, cross A, crossing between genes Y and W, where this is the male YYWW and this is female YYWW, brown body, red eyes and yellow body, white eyes. In the F1 generation, when it is selfed, after the result, he was expecting 9:3:3:1, but results were not just like the above. The results were different from the above. What was the results? But Morgan found that 98.7% combination was parental. Basically, parental combination, percentage of parental combination was higher than the Mendelian inheritance. It was almost 98.7%, while only 1.3% combinations were recombinant type. It means new results were only 1.3%. He concluded that the genes of body color and the genes of eye color were linked genes. So 98.7% recombinant combinations were found. It shows the linkage. It means this is the deviation. This is the deviation, and this deviation is because of the linked genes. You can see the figure. This figure is taken from your book. You just need to understand only one thing. These two spots. These are the two genes, yellow and white, and this is the wild strain where brown bodied red eyed female wild type and yellow bodied white eyed female. When it is crossed, parental type was 98.7% and recombinant type was 1.3%. These are the gametes, and you can see the results in F2 generation, where parental type was more than 98%, while the recombinant type was less than 2%. What about the crossing B? Crossing between gene W and M. In another experiment, white-eyed and short-winged female Drosophila was crossed with red-eyed and large-winged Drosophila. Here, dominant wild-type alleles are represented with plus sign in superscript. So you can see Morgan found that the results were almost similar to the Mendelian. So in the cross B, results were similar to Mendelian dihybrid cross. It shows that this is the non-linked genes. Morgan found that the results were almost similar to the Mendelian dihybrid cross. When two genes in a dihybrid cross were situated on the same chromosome but far distant positions, then the proportions of the parental gene combination was only 62.8%, while the non-parental type or recombinant type was 37.2%. It means distance matters. What is the distance between two genes? What is the reason? The genes of this trait were at a large distance on the chromosome. You can see the figure here. This is the figure. This is W gene and M gene, and the distance is very large in between these two. So the parental type is 62.8% and recombinant type is 37.2%. You can see the cross B, crossing between genes W and M, WWMM. This is white and miniature wings, while the W plus X, M plus X. This is the wild strain, and he received almost 9:3:3:1 type of results. What was the results? But Morgan found that 62.8% combination were parental, while only 37.2% combinations were recombinant type. He concluded that the genes of eye color and the genes of wing size were not linked genes. So 62.8% parental combinations were found. It shows the Mendelian inheritance. You can see the figure here. This large distance is responsible for the Mendelian results here, 62.8%. Now, some important facts about the linkage. Linkage, a physical association of genes on a chromosome. What is recombination? The generation of non-parental gene combination is called recombinant generations. Morgan found that even when genes were grouped on the same chromosome, some genes were very tightly linked showed very low recombination, while others were loosely linked, showed higher recombination. So attachment, type of attachment is also important. Tightly linked, it means lower the recombination, and loosely linked means higher the recombination. The genes white and yellow were very tightly linked and they showed only 1.3% recombination. The genes white and miniature wing showed 37.2% recombination, hence loosely linked. There was a scientist Alfred Sturtevant. Alfred Sturtevant used the frequency of recombination between gene pairs on the same chromosome as a measure of the distance between genes and mapped their position on the chromosome. So we can prepare a gene map by using the distance of the genes. Today, genetic maps are exclusively used as a starting point in the sequencing of whole genomes, and it is used in Human Genome Project (HGP). You can see the important figure here. This is chromosome, this is chromatin, this is the nucleosome model, histones, this is nucleosome, this is the DNA helix, and these are the sequences on the DNA. Now, genetic mapping. What is genetic mapping? It is a way to identify exactly which chromosome has which gene and exactly pinpointing where that gene lies on the particular chromosome. Basically, it shows the position and place of the genes on the chromosome. What is the perfect position and what is the sequence. Mapping is also act as a method in determining which gene is most likely recombine based on distance between two genes. And the unit of distance between gene is centimorgan. It is shown by small c capital M. This is the genetic map. You can see cytogenetic map. This is the physical map, and this is the DNA sequence. Although genetic mapping is beyond your syllabus, it is the part of higher classes, so you will study all these things in detail there. Now the next question, or you can say next part of the topic, that is parallelism in chromosome and genes. Both the chromosome and gene shows some sort of parallelism in there. It was proposed by Sutton and Boveri in 1902. They recognized a close parallelism between the transmission of factors identified by Mendel and the behavior of chromosome during meiosis and fertilization. What are the Mendelian factors? Alleles of a factor occur in a pair. Similarly, chromosome also occur in a pair. Similar or dissimilar alleles of a factor separate during the gamete formation. At the time of gamete formation, alleles or factors are separated. Similarly, the homologous chromosomes separate during the meiosis. Mendelian factors can support independently and the paired chromosome can separate independently during meiosis. But the linked genes in the same chromosome normally do not assort independently. Important postulates of parallelism. What are the important facts about the parallelism? Both the chromosome as well as Mendelian factors are transmitted from generation to generation in the unaltered form. A trait is separated by only one Mendelian factor inside a gamete. A gamete similarly contains a single chromosome out of a pair of homologous chromosomes due to the meiosis that occurs before the formation of gametes. Similarly, an organism has a specific number of chromosomes, such like the human being has 46 chromosomes in its cell. The somatic cells are generally diploid, having chromosome in the pair called homologous pairs. On the other hand, the two chromosomes of each homologous pair resemble each other in the morphology and genetic content. They are derived from the two parents through their gametes. It also contains two Mendelian factors for each character, and the factors come from different parents through their gametes. Some important discoveries in the genetics are given here. 1831, Robert Brown discovered the nucleus. In the 1848, Hofmeister discovered the nuclear fibers. 1865, Gregor John Mendel, Laws of Inheritance was proposed. Friedrich Miescher, 1869, he discovered the nucleic acid DNA and he gave the name nuclein. 1879, Waldeyer, he discovered the chromosomes. In 1882, Fleming, movement of chromosomes. 1887, von Benden discovered the meiosis. In the 1900, rediscovery of Mendelian inheritance by three scientists. And 1953, Watson and Crick, and they proposed DNA double helix model. Fleming discovered the mitosis, and Farmer and Murray discovered the meiosis. Now we will discuss about the sex determination. What is sex determination? What do you mean by the sex determination? Basically, it is a biological system that determines the development of sexual characteristics in an organism. Autosomes controls the somatic traits, while allosome controls the secondary sexual characters. And it was discovered by McClung in Locusta. Locusta is the genus of grasshopper. Hanking, he traced a specific nuclear structure during the spermatozoa of Locusta. An insect, 50% of the sperm received these specific structures where 50% sperm did not receive. Hanking gave a name to this structure as the X body. X body of Hanking was later on named as X chromosome. It is given the X chromosome. Now, classification of sex determination. It is discovered by Natty Stevens, 1903 in mealworm. There are following type of sex determination can be observed: XXXY type, XXX0 type, and ZZZW type. So come to the first XXXY type. It is further subdivided in two categories: XX female and XY male. This is found in human beings, in the Drosophila, in the Coccinia and Melandrium. Coccinia and Melandrium are the plants, while the human and Drosophila are the animals. So female is homogametry here. So homogametic female is seen in the type, and while XX male and XY female type, male is homogametry, species, Reptilia, Aves, and Fragaria. Fragaria is the example of plant. So in the XX male and XY female type is also known as ZZZW type of the sex determination, where ZZ is male and ZW is female. It is applicable in birds like cock and hen. In birds, sex is decided by female individual. So basically when we talk about the cock and hen, then the sex is decided by the female, not by the male. And the next and important is XXX0 type. In some organisms, XX is female but X0 is male. It means male will have, or you can say male will possess a one less number of chromosomes than the female. Example is Locusta, grasshopper, cockroach, and ascaris. These are the some important examples that shows the XXX0 type of combination. A question is given here: there is an insect in which XXX0 type of sex determination is found. If the female insect has 20 chromosomes, then find out the number of chromosomes in male. Answer is 19 chromosomes, because male has one less number of chromosomes than the female. You can see this is the diploid nuclei. Female, it has the diploid condition, while this is the 0. It will form two type of gametes XX, while male will form only one type of gamete that has the X, while another gamete will not show this. After fertilization, this is the zygote XX and this is X0. Sorry. Sex determination in human. In human beings, XX female and XY male type of sex determination is found. So this is again important point. What type of sex determination is found in the human beings? XX female and XY male type of sex determination. Human sex is determined by the presence of or absence of a Y chromosome with a functional SRY gene. Once the SRY gene is activated, cells create testosterone and anti-Mullerian hormone. Basically, SRY gene will produce testosterone and AMH, anti-Mullerian hormone, which typically ensures the development of a single male reproductive system. A sex-determined region is found on Y chromosome that has X gene. So this is the presentation of gene here. As I have already asked in the test, S or Y, small in letter, and similarly italics, which is also known as TDF, testis determining factor. Human has 46 chromosome as a diploid number. The male has 44 plus XY and female has 44 plus XX chromosome. And this figure shows each and everything here. Father is with XY and mother has XX. And when you will see the cross, there are the 50% chance of girl child while the 50% chance of boy child. Not in Indian society, females are blamed for the birth of girl child, but in real, male is responsible for this means. Male is the biological father of the child. Now, sex determination in honeybees. Although this is beyond your syllabus, so we will not talk about this. Now come to the next, pleiotropy. What is pleiotropy? Pleiotropy word is derived from Greek words pleion means more and tropos means way. Pleiotropy occurs when one gene influences two or more seemingly unrelated phenotypic traits. It means one gene is influencing the two characters which are phenotypically not related. Such a gene that exhibit multiple phenotypic expression is called pleiotropic gene, and this phenomena is called pleiotropy. A single gene can exhibit multiple phenotypic expressions. Such gene is called pleiotropic gene. And the mechanism of pleiotropic in most cases is the effect of a gene on metabolic pathways which contributes towards different phenotypes. You can see this is the single gene. The name of gene is beta globin gene mutation, sickle cell anemia gene. Now what will you see? By the effect of this gene, you can see the pleiotropy. There will be the phenotypic character of the eyes. It will show the blindness, liver failure, and similarly heart attack. It means it is a multiple traits, single gene and showing the different effects. Now there is a disease that is PKU, phenylketonuria. This disease is inhuman is an example of pleiotropy. It's an inborn error of metabolism. So it will cause the metabolic error and is also inherited as the autosomal recessive traits. This disease is caused due to mutation in the gene that codes for the enzyme phenylalanine hydroxylase enzyme. The name of enzyme is phenylalanine hydroxylase that converts the amino acid phenylalanine into tyrosine. In the absence of this enzyme, the phenylalanine is converted to PPA. The name of PPA is phenylpyruvic acid, which is accumulated in the brain and it will make the person full. What are the phenotypic expression characterized by phenylketonuria disease? Mental retardation. So mentally person will be weak, reduction in the hairs, reduction in skin pigmentation and excreted through kidney because it shows poor absorption. What is the biochemistry of the phenylketonuria? You can see here. If the phenylalanine is present and in the presence of phenylalanine hydroxylase it is converted to tyrosine. When this enzyme is absent, then tetrahydrobiopterin will convert to this one and it will change the biochemistry. In the absence of phenylalanine hydroxylase enzyme, the phenylalanine is converted to PPA, phenylpyruvic acid, which is accumulated in the brain. Now, a single gene product may produce more than one effect. It is seen in the starch granules in pea plants. So what will you see? That Starch synthesis in pea seeds is controlled by one gene. It has two alleles, capital B and small b. So starch is synthesized effectively by capital B homozygote, and therefore large starch grains are produced. So if the both the alleles are dominant capital B, then the size of the starch grain will be large. And small b homozygous condition has less efficiency and produce smaller grains. And after maturation of the seeds, capital B seeds are round in the nature, or you can say round in the phenotype, and BB is the wrinkled. So heterozygous BB produce round seed, and so B seems to be the dominant allele, but the starch grains produced are intermediate size. It is showing the codominance. In starch grain size is considered as the phenotype, then from this angle the alleles show incomplete dominance. Again you can see the pleiotropism. Usually one gene controls one trait, but when one gene controls more than one trait, it is the case of pleiotropy. B gene, we are talking about the grain size of the starch in pisum. Pleiotropy, shape of the seed, it will also affect the starch grain synthesis. The type of starch grain will decide the shape of the seed in Pisum sativa. Next, you can see the mutation. What is mutation? Mutation is a phenomena which results in sudden alteration of DNA sequence and consequently results in the change in the genotype and phenotype of an organism. The mutation was first seen by Cartwright and in Anconship, small leg. Cartwright was not a scientist; he was a shepherd, and discovered by Hugo de Vries, and he proposed the mutation theory in 1901 to 1903. So what will you see? Suppose this is the DNA and DNA copying is going on, then this is original. Suppose this is the purple nucleotide sharing with green one. If it is replaced by yellow, then it is both the side of base pairs will be affected. This is the mutant copy and this is the correct copy. In addition to recombination, mutation is another phenomena that leads to the variation in DNA. Regions of mutation. What are the basic regions of mutation? There are 4 chances of the region of mutation. First is deletion. If any gene or allele or a sequence of DNA is deleted. Duplication, presence of duplicate copies. Inversion, it means sequence is inversed. And translocation. So what is deletion? It is a mistake in the DNA replication process which removes nucleotides or a segment of DNA from the genome. And a deletion mutation can remove a single nucleotide or entire sequence of the nucleotides. You can see the sequence here. This purple color area is removed from this chromosome. This is before deletion, and after deletion the size is shorter. Duplication or insertion. This is the single sequence. It is present in duplicate copy. You can see the 2 copies here. So in this type of mutation that involves the production of one or more copies of a gene or region of chromosome, a segment of DNA is result in alteration in chromosomes. Third is inversion mutation. And inversion is a chromosome rearrangement in which a segment of a chromosome is reversed end to end. You can see this is the sequence of the genes. In the first chromosome, green, orange, light blue and purple. Here the sequence is inversed here. Orange is on the top, here orange is down, and blue is in the lower position, this is at higher position. And fourth is frame shift mutation. What is frame shift? You can see a frame shift mutation is a type of mutation involving the insertion or deletion of a nucleotide in which the number of deleted base pairs is not divisible by 3. although this definition is not given in your book. Change in the frame of nucleotides. Since genes are located on the chromosome, alteration in chromosome result in abnormalities or aberration. Chromosomal aberrations are commonly observed in cancerous cells. You can see here, this is ATG, GAA, GCA, CGT. And this sequence is producing these type of amino acids, methionine, glutamic acid, alanine and glycine. As the sequence is changed here, what will you see? ATG similar to the first one, and this is AAG. This G is deleted. After the deletion AAG, this is the lysine, and again you can say CAC, this is histidine, and last two nucleotides are left behind, and this is incomplete sequence. So frame shift mutation is causing the difference in nucleotides. Mutagens, these are the physical, chemical or biological agents that changes the genetic material, usually DNA or an organism. So basically mutagens may be physical, may be chemical or biological. It increases the frequency of mutations above the natural background level. Types of mutagens, physical mutagens, it includes the radiation, temperature and humidity. DNA, reactive chemicals, base analogues, intercalating agents, metals, biological agents. This is the type of mutagens. Here you can see the physical, it includes radiation and heat. Chemical mutagens, it includes base analogues, alkylating agents, intercalating agents, deaminating agents, metal ions. And biological mutagens include viruses, bacteria and transposons. So this is the type of mutagens. So dear students, we have discussed here a lot. Here we have discussed the linkages, parallelism in the gene and chromosome, mutation, type of mutations, regions of mutations, and the mutagens, all these things. So if you have any problem, you can ask to me. Till then, good day.